much of time, let us get into the evolution of Newcastle disease virus and the novel approaches to control. So this is the topic which I have chosen to speak in this particular forum. So as everybody knows, it's a 100-year-old disease, a century-old disease, which we are trying to talk even today about the massiveness which is causing a disaster in the poultry industry. Around the world, it is nothing related to only the Nepal, but around the world. But apart from that, there are other challenges which also has caused the industrial damage in the recent past also, which we have to also think in control measures apart from that as a differential diagnosis or whatever it is. I'll try to cover on a wholesome this thing where we have to interfere and where we have to differentially diagnose and we have to control these particular viruses. So as we have seen, there is a history of almost 90 plus years of control measures for this particular viruses with the vaccines. Always we think that the vaccines are going to help. Even these vaccines are being used as a therapeutic usage in the poultry industry. That is the practices which is happening in the poultry industry today. But as Paul Ehrlich has coined this particular thing like vaccines as magic bullet. When he coined this word as vaccines as magic bullet, within a decade, he withdrew this word because of several things which he never imagined that it is a magic bullet as a whole. Okay, so in the present presentation, in the sense, I will be focusing on these particular areas in detail about the reasons for endemicity of the century-old diseases like ND and also some of the other related infections and how the evolution has happened with the NDV genotypes. There is a lot of confusion happening. Some people will be calling it as genotype 7, some people will be calling it as 13 or some of the new. What is the changes which has happened? What is the confusions? I will try to clarify those things. And definitely the control measures with the vaccines over the past and the present, how the things are changing and what should be the goal of the ND vaccination. That is one more topic which I would like to cover, and the concepts of early protection and a long-lasting immunity. Early protection in the broilers or in the long-lived birds and the long-lasting immunity, how we have to control all those things. And last but not the least, vaccines or the vaccination failure, which we need to address. So before getting into that particular thing, let us try to understand some of the things present with the present pandemic, which also gives a lot of lessons for us to understand about some of the diseases and the changes which is happening with the disease and the control measures. So which gives a lot of thing with the COVID-19 pandemic, which is happening in the present. Initially, when the COVID-19 pandemic happened, so people started blaming the poultry industry in the initial phase. So the poultry industry crashed. This happened in India too, apart from the several other neighboring countries. Poultry was blamed for that, but people forgot to understand that the COVID viruses or the coronaviruses, which is derived from the mammals, will not, and the mammals and the birds are completely different, which causes mammals are derived from the bats, which circulate among the domestic animals and are the wild animals, and we can then can infect the human beings. That is how the changes happen with the mammalian coronaviruses, with the SARS or the COVID-19, which is happening. So which affects the birds completely, it is different. It is the presently which is happening in the birds, it is the infectious bronchitis virus, which belongs to the gamma coronavirus. Here I would like to stress here some of the things which is related. See how the viruses evolve. The viruses keep on mutating, especially related to the RNA viruses. So they become endemic if we are not able to properly control that and eradicate it in the initial phase. It gets established and causes this thing. That is the one important. And the changes happening with the virus, you can know with the COVID, several changes happen with the UK strain or the South African strain or the Australian strain. This is how the things are changing. And To coming to the control, initially it started with the biosecurity. The movement of the people, transport industry was completely blocked. That is how you control the virus, this thing. That is what we need to adapt in our industry too, which majority of the things we try to compromise in the poultry. And the disposals which is happening, cleaning and disinfection, and this is how the things happen with the, which we have to learn in the disease control measures. And the next thing is the comorbidities. In humans, the comorbids like the diabetic patients or the BP patients or some other diseases, people are affected more, and there was death in those people. Like in poultry, we can see several immunosuppressive diseases. Even the feed, which is the quality of the feed, matters in causing a lot of immunosuppression issues, which leads to a lot of aggravation of the diseases, even sometimes the vaccinal reactions also, which we need to understand. Then last but not the least, the vaccine efficacy. We are talking about a lot of live vaccines of coronavirus in poultry, which has been rampantly used around. That is how, even with the live vaccines of the Newcastle also. But in humans, if you take, there is no live vaccines for coronavirus. It can change and mutate and cause more disasters. That was this thing. That is the lessons which we need to learn in some of the viruses. If it is a mild virus, definitely we can use it. But if it is changing, we need to intervene. And the inactivators or the recombinant vaccines which has emerged in the industry, we need to understand those things too. Last but not the least, herd immunity is very important to control any disease. So to limit the spread. So how to do all those things, how to adapt to the disease control measures in poultry are especially related to Newcastle, I'll try to address in this particular forum. Yeah. So first of all, I will try to address about the reasons for endemicity of this century-old disease around the world. So if you see the endemic diseases prevalent around the world, the top 5 diseases comes into the category of RNA viruses, which has an important character that RNA viruses keep on mutating. They keep on changing for it to establish and to cause more disaster. That is what is happening with the RNA viruses. We need to understand, especially related to the avian influenza or the Newcastle and the infectious bronchitis virus. Majority of the countries neglect infectious bronchitis virus, which is, if you take out the HPI because of the culling program, infectious bronchitis definitely is the top 1 disease in poultry too, like humans present currently with the coronavirus. So let's concentrate on the Newcastle disease. If you talk about Newcastle disease, I always compare that with the high pathogenic or the low pathogenic viruses. We can categorize in coming slides. I will try to categorize those things too. Okay, so if you talk about the characters of these viruses, especially related to the influenza and Newcastle, where always I find it more similar in some of the clinical signs and in some of the clinical picture and the postmortem lesions, majority of the time. But there is a lot of differences in the character of the virus, especially the virus family. If you see, the influenza belongs to Orthomyxoviridae and Newcastle, it is a Paramyxoviridae. When it comes to the genetic material, it is a segmented RNA virus, influenza. When compared to the the Newcastle disease, it is a non-segmented, single-stranded. The mutations will not happen that much compared to the influenza. If you see, even influenza, you can find several serotypes around the world, which is prevalent and keeps on changing. The serotypes keep on emerging out. But in Newcastle, till today, though there are several genotypes, it is only single serotype. That's why we are able to control this particular virus compared to the influenza viruses. Okay, so coming to the virulence, it depends on the fusion protein cleavage for the, this is opposite. In influenza, it is the hemagglutinin cleavage site, and in the Newcastle disease, the fusion protein cleavage site is very important so that it becomes virulence. This is one mistake which has to be corrected in the during the presentation. So when it comes to the pathotype, if it is influenza, the high pathogenic avian influenza or the velogenic viruses are very similar lesions. Hypopathogenic means it is more related to the H5 or the H7 isolates, which causes the bird flu in the poultry. So that is where the LPI I always correlate with the lentogenic viruses of Newcastle disease virus, which is a non-H5 or H7, which is several types. And as I told, there is several serotypes in influenza and a single serotype, and no live vaccine in influenza because of the mutation capability, and it can lead to a new emergence of variants. This is banned. That is not being used in any of the species. But if you take Newcastle, we are today using the lentogenic as live vaccines, and even in some of the Asian countries where the Newcastle disease is endemic, we are getting good results with the mesogenic strains too, like R2B or the Mukteshwar strain. Okay, so the clinical signs and PM I have already explained. So about how it correlates with the ND. Let us try to learn more about its pathogenesis now. If you talk about the virus pathogenesis in this Newcastle or the influenza viruses, the spread is via the aerosol or the fecal-oral route. That is the common route in both the cases. Here in Newcastle, it is the fusion protein gets attached to the epithelial cells and gets into the cells and starts damaging the upper respiratory tract and the GI tract. That is the initial phase of entry into the cells and replicates and damages the upper respiratory and the GI tract. And you will get a lot of tracheal lesions or the enteric lesions in these cases in the initial stage. And it is very common in the lentogenic or the low pathogenic avian influenzas. So these viruses are limited to the upper respiratory and the GI tract. They are not entering the other tissues as such in the LP or the Newcastle disease. But there will be reinfections from which is shedding into the environment from the upper respiratory secretions or the GI, the fecal route that will be spreading and recirculating the viruses into the environment, and new cases will be happening in the next floors. But in some of the viruses like the velogenic Newcastle or the high pathogenic avian influenza, it enters the systemic circulation, enters into the system and can affect all the organs in the system, including the brain. You can get a lot of nervous lesions or the torticollis, which we call the clinical signs that we can get. And the virus is found in most of the tissues within a span of 22 to 44 hours. That is the capacity of these viruses to spread and cause high mortalities or high infection. It can damage all the systems. So if you see, these shedding can lead to, once again, secondary viremia, and the infections can spread. And these viruses, that is the velogenic Newcastle disease or the hypopathogenic avian influenza, they are called the catastrophic diseases of poultry, where you can, in an unvaccinated flock, you can see 100% mortality within 10 days. That is the capacity with the velogenic Newcastle disease virus or the hypopathogenic viruses. So, but in a vaccinated flock, definitely there can be the protection, but it depends on the immunity levels, which I will be talking in the later stage. Okay, but if it is a lentogenic or low pathogenic avian influenza, the mortalities may not be very high. It can be limited to less than 5% of the mortality, but the secondary complications can lead to a lot of production issues. So that is where we need to differentiate. This is one of the table which shows what is the molecular basis of these virulence or pathogenicity of immunogenicity. In this, I have tried to compile in this particular taking examples of the Newcastle disease strains. Like if you see up to Lasota, from the V4 strain or the Ulster strain or VH or F strain, all these VGGA strain, all these strains, if you see, they are classified as apathogenic or lentogenic viruses, which has low ICPI values, that is intracerebral pathogenicity index, which is done in SPF chicks by inoculating it to the brain. Okay, so this has a low virulent capacities based on the the ICPI values, but there are certain strains, that is the Mukteshwar, Komaro or the Roykin, which has a slightly higher ICPI values, are classified as mesogenic strains. And there are certain velogenic strains like the CA108 or the Texas or the genotype 13, which is prevalently in the Asian countries. This has a high ICPA values more than 1.7 or 8. So up to 2, it can happen. They are classified as velogenic. If you see, the virulence I have categorized based on the strains here. The virulence increases, and even the immunogenicity increases with the increase in the virulence. So if you see, these strains are more immunogenic if you are using in the for the protection, homologous protection, compared to any of these strains. That is what I mean to say by this. So when it comes to the virulence, also are related to the fusion protein cleavage site also. That is, the F0 is divided into F1 and F2. Then only the virus starts replicating in the system. So if you see the lentogenic viruses in the fusion protein cleavage site, there are repeated basic amino acids like the arginine. Okay, so this has been recognized by only trypsin-like enzymes, which is present in the upper respiratory and the GI tract. When these enzymes recognize this particular amino acid sequence, there is a cleavage and the virus multiplies here. And in mesogenic or velogenic, the basic amino acids are repeated, which is not repeated in the lentogenic viruses. It is repeated amino acids where any proteases present in the system can replicate. So that's why if you see, the lentogenic viruses are given in the intraocular route or the oral route, the drinking water. But the mesogenic strain vaccines are given by intramuscular route, where it can replicate in the system and it can be recognized. That is what happens. The same thing happens with the influenza viruses with the HA0 protein cleavage site, which is very similar to the low pathogenic or very similar to the lentogenic viruses, and the high pathogenic is very similar to the velogenic viruses, where it can replicate in all the organs of the body. That is what is the difference. You can see this is the non-H5, H7 isolates, which is related to the upper respiratory and the GI tract. Okay, so this is how it has been classified. If you see, compared to the Newcastle, the influenza viruses have a very tremendous changes can happen because of the complex diversity, because of its genome. It's a segmented genome where a lot of reassortment can lead to different types of pathogenesis and the changes happening. If you see the low pathogenic avian influenza virus, that is H9N2, you can see it has been derived from so many different hemagglutinin viruses derived from which has affected different species. Okay, like the H5, H2, H3 and all. So that is how the changes happen, which is more virulent compared to this thing. The same thing, if you see, then coming to the epidemiological features in the Newcastle ND endemic countries, if you see, there is no stamping out policy. The Newcastle disease are not reportable diseases in most of the countries which is having velogenic ND. That is not a reportable disease. That is how it has established, and a lot of things are happening with this. And the second thing is with the vaccines and the vaccination procedures. And the third is the high density, which we cannot control in the present scenario. It is increasing. This is one of the Google map where a lot of poultry is happening. If you find any Newcastle outbreak is any of the farm, you can see how it can spread and lead to disasters. And biosecurity protocols and lack of surveillance programs. And last but not the least, untreated poultry rectal disposals, which is happening into the agriculture or this thing, which can lead to the persistence of this particular organisms. Okay, so next, let us try to understand the epidemiology of transmission, how it gets. As I already told, it is the fecal-oral route or the respiratory, it can get transferred. If a bird is affected, it can, within the infected premises, it can secrete through the excretions or the fecal shedding and spread to different these things. And it cannot be transmitted vertically. This virus is not very vertically transmitted, but it can be transmitted by the other horizontal routes. By contaminated eggs on the surface, it can definitely transmit, not a vertically transmitted disease. And To the premises, how it is going to come, it is through the vehicles or the peoples, the vaccinator crew, or some of the rodents. Even the dogs can be a transmitter. The virus can persist in the dogs for almost 6 months. That has been proven. The flies, rodents, all those things. The contaminated feed and water, especially in the broilers, where if there is from the infected the litters are disposed next to the farm. You cannot expect the next flock will not be affected with the same litter material which is being disposed next to the farm. So that is what is happening, and the disposal is a big problem which is happening. Or the live poultry or the dead birds with disposals which is not happening properly can be a very good source of transmission. Lastly, the wild birds, which is a major source of transmission of majority of this virus. Almost 250 species of birds are affected. It includes even influenza viruses can affect these wild birds, and it can be a major transmission source. So definitely, if you see, when I talk about the migratory bird, which is spreading the infections. So this is some of the routes which has been studied about the migratory birds, especially when they travel during the winter seasons from the cold countries to the warmer countries. So where the diseases are being transmitted to different countries. That is how, this is some of the routes where some of the Asian countries and the African countries are affected. Okay, so I'll give you one of the example how the recent H5N8 has spread across the world in the last pandemic, that is from 2020. It was recognized in the Saudi Arabia in February 2020. The first time it occurred outbreak in the wild birds. From there, it started the disease outbreak was seen in the Russian and the Central Asian countries. Then in the recent November, December to till date, a lot of European countries are affected with this, and they have reported this particular disease happening. Then it moved to the Japan and South Korea, China, India, and some of the Asian countries where it caused a lot of. This is how the route of transfer happened with this particular virus, which we need to understand how the migratory birds can transport these particular viruses. And recently, human cases has been reported in March 2021, but none of them had a human-to-human transmission, which we need not worry in the present scenario. But we have to keep a track on such sort of viruses. Coming to the clinical picture, what we can see with the outbreaks of the Newcastle disease and how to differentiate with the other diseases, which is very important. Most of the time, the Newcastle diseases start, as I told, with the upper respiratory lesions like the tracheitis, the catarrhal tracheitis, or the proventricular lesions, or the intestinal regions with the cecal tonsils getting affected, or the patches. And even sometimes the vaccinal reactions which happen leading to the air sacculitis, perihepatitis, or the pericarditis that is very commonly seen in such sort of cases. Apart from that, definitely production drops in the laying flocks, and a lot of misshapen or the shellless eggs can be seen. This is more related to the milder strains of viruses which we very commonly see. So if you see the next particular clinical signs, it is more related to the velogenic type of viruses, where the nervous strains like torticollis can be seen in majority of the long-lived birds as well as the broilers too. Okay, so if you see, this is some of the clinical signs which has been in the video, which you can observe the torticollis lesions. But all these cases has to be differentiated from other disease clinical signs too. Like if you see, these are all we have seen in such sort of lesions in vitamin deficiency cases, that is especially related to the vitamin B12 or vitamin E deficiency. Even in the day-old hatchery, we have seen certain torticollis issues in the first week, but in the hatchery, when we got back, we saw some of the lesions in the pipid embryos where the lesions of the skins were also found, apart from the nervous signs, which we differentiated, we isolated. AE in those cases. The vaccination errors, which happen with the subcutaneous by damaging the blood vessels, or if the vaccination happens much near to the head, which always leads such sort of lesions, so which we need to differentiate. Okay. So, and sometimes even some of the bacterial infections happens to the ear canal, that is the otitis with the bacterial infection can enter into the nervous system, the brain, and cause a lot of lesions. Even some of the bacterial like the coryza or the fowl cholera or ORT can cause that sort of torticollis issues, which recently has been published in several papers. Okay, so this is with the middle ear infections. So the next clinical signs which we commonly see is the consolidation of lungs with a lot of hemorrhages and those things, and petechial hemorrhages on the ribs, or some of the petechial hemorrhages on the fat tissues or the phlebitis. Most of the time, such sort of lesions are definitely part of the Newcastle, the velogenic viruses, but we need to differentiate this with the high pathogenic avian influenza and also the fowl cholera. Fowl cholera lesions also gives very typical lesions like the petechial hemorrhages on the rib cages or the consolidation of lungs, even sometimes necrotic foci. So such sort of lesions which we need to differentiate and take the help of the lab. And the oophorytis cases, which you can see the lot of hemorrhagic follicles with misshapen, we can see in the Newcastle disease definitely, but we can see in other cases of the influenzas as well as the bacterial infections like fowl cholera as well as salmonella, so which we need to differentiate. So this is some of the diseases which we need to differentiate. If it is a mild form, we have to differentiate with the clinical signs of these which I have listed here, and the severe form with the high pathogenic or some of the tracheitis with the ILT, all those things. And especially the acute fowl cholera gives very typical lesions of the some similar to the high pathogenic or the Newcastle disease. Yeah. So from these samples, to differentiate all those things, we have to adapt certain molecular diagnostic tools to identify the viruses or the complexes which happen with the different bacteria or other viruses, and the complications which happen. And also we need to know the strains which are prevalent around the world. We are today talking about genotype 13, which is prevalent, or genotype 7I, which is prevalent. I will talk about those things. We need to differentiate. For that, molecular tools are very important. Like we can take the samples, the clonal swabs or the tracheal swabs and some of the tissues. We can extract RNA from that. We can go for reverse transcriptase PCR and identify the virus if with the specific primers used. If it is positive, we can go for sequencing of that particular fusion protein and we can get the strains related to this thing. So, and also we can also know what all other bacteria or the viruses are involved in this particular complications happening. Okay. So we can take these lessons from the recent screening with the humans with the COVID virus, really with the real-time PCR. Okay. So the next is the evolution, how the genotypes have evolved around the world. You can see the Asian countries are affected with the genotype 13. Okay. Let us see how the pandemics of ND has happened since its inception into the recognized in 1926 with the in England. It was more related to the lentogenic viruses of Newcastle disease, which was categorized in the present scenario. It has been categorized as genotype 1, 2, and 4. So this is what the initial, which was more belonged to the lentogenic or the mesogenic type. There was no velogenic during that time, but the viruses started changing on its own and started emerging with the new genotypes emerged in different phases. If you see in the 1960s, it was more related to the genotype 3, 4, 9, and 10. Later on in 1980, in the Middle East, it was more of a pandemic with the genotype 6. But of late, after 1984, for almost 3 to 4 decades, it is more problem of pandemics with genotype 7, which is the most variable genotype in the present, which is circulating around the world. Okay, but majority of the genotypes are falling in the category of 13 in the present scenario. But that is not the end. If you see the latest outbreaks which is happening in some of the countries like Indonesia, Israel, and Pakistan, it is genotype 7I, which is classified as 19, which is being circulating very fast in some of the countries and causing more outbreaks, which we need to track. Some of the cases have been found in India, but it has not spread very fast. We are being able to control those things. We don't have much issues with 7I, but we have with the 13. If you see some of the how the diseases have spread with the recent pandemic, especially with the genotype 7I, if you see, it started in 2010 in Indonesia, and it has affected Pakistan, Israel, and some of the Middle East countries. And it has even spread to the European countries in 2019, 13. So that is how the spread is happening around the world. If you take 13B, we are being affected with the 13B and C isolates also, which has spread to some of the Asian countries and some of the European countries too. It is the other virus is 7H, which is being spread from the Indonesia to the African countries, where it is mutating and spreading across in Africa. And from Malaysia, 7I, which is spreading in some of the Southeast Asian countries, which has been spreading there, but it is not prevalent in some of the other countries of Asia. So we need to track the genotypes in that way to derive these things. And today there was a lot of confusion with the classification of these genotypes. Why this happened, There was an earlier classification based on the fusion protein sequence where it was partial sequencing. They were classifying based on the partial sequencing, short sequences of this, and they were classifying. In that, the 7, if you take the genotype 7, there used to be different subgenotypes like 7A, B, C, D up to H or I. It was being classified in the earlier cases. Okay, that is how, and it lacked a lack of uniformity in the nomenclatures among the scientists. They were not able to derive a proper this thing. Later on, there was a publication which happened where they reclassified based on the complete fusion protein gene analysis, and they derived a different sort of classification based on these parameters. That is, if there is 10% or more changes in the fusion protein gene, they classified that as a different strain itself, like 7B, which was classified as 13, 7I as 19. That is how the classifications, they gave a new terminology or the new number to that particular strain, which has more than 10% variations. That is how now presently we have recognized more than 19 genotypes. There are even 21 also in some of the publications, but it is not a concrete, this thing has not been established about the last 2 strains. Okay, but the avirulent or the low pathogenic strains are still remaining in genotype 1, 2, and 10. They have not reclassified those things, and there is no change in those cases as such. So if you see about the genotype 13, people are talking that this virus 13 was seen after 2013 or 14. That is what, it is a new virus like that. People are talking much about this. But if you see, most of the ancestral strains of genotype 13 was first recognized in 1982 in the cockatoos, which was recovered from India. That is how it has been published. You can see that was the old isolate. But if you see some of the publications, all these authors listed here have published their data about the recent Newcastle disease, which happened before 2015 as genotype 7. But these publications, whatever the genome which has been submitted, now this is automatically coming under genotype 13. So that is how the changes has happened in the classification, which has led to the confusion. So no need to much bother about this particular strains. So if you see, which is being presently circulating in our country and some of the Asian countries, in the neighboring countries also, like Nepal also, we have certain isolates we've been classified. If you see, they are being classified as 7B genotype 13. So these are all the recent isolates in which has been classified like 2013, 2019 also. You can see here some of the North Indian isolates. So this is how the classification. And the low pathogenic or the live vaccines comes in this category, okay, which has been classified in this particular phylogen. So if you see some of the isolates which I have taken and analyzed about the recent, in the sense which has been submitted to the gene bank with the Nepal isolates, I have found only the old isolate which has been submitted, which is classified as genotype 13. But recently, there are people talking about some of the 7C or something, which we need to know what exactly is that, whether it is a 7I, which is circulating in some of the neighboring countries, or it is 13, which has emerged out in a new form, which is what we need to analyze. We have to take the genomic analysis in a better way. Okay, so the next topic which I'm going to talk is about what is the vaccines developed for the last 90 plus years to control this particular virus, and what are the different strains and how we are able to control with these strains. Let us try to understand how the changes has happened with the vaccines. So if you take the 90-year-old story of the vaccines used as control measures for Newcastle, the initial control started with the Mesogenic strain, such as Roykin strain, Herd strain, or the Mukteshwar strain. In different countries, they were having different Mesogenic strains, which was used for the control in the initials. But it has its own advantages, disadvantages, which I will be talking in the later slides. It led to the identification of the lentogenic strain with the milder forms. Then came the inactivated vaccines, then came with still the virulence was persistence, which was affecting some of the cases, leading to vaccinal reactions. Then these cloned vaccines or the recombinant vaccines were derived to reduce the reactions which was happening. So that is how the industry is working in identifying a better vaccine strains in controlling these things. Let us try to understand about some of these strains in this particular form. If you see the Mesogenic strains, which was first introduced in the 1930s, first identified by Herd strain, first identified strain was the head strain, that is Herd strain by Ayer and Dobson. In England, they identified this and they started using as a vaccine strain. Seeing that in US, Budet, who was called as Red, his name this thing was called as Red. So he isolated almost 105 isolates. His lab was having almost 105 isolates of Newcastle from different outbreaks around the country. So from those, he identified Roykin strain to be a best strain and he introduced for vaccines in those at that time. Later on in India, Ayer came back and identified the Mukteshwar strain that is being named as R2B, okay, which was used in India, and the Komarov strain and some of the Palestinian, this thing. These were from different countries they had identified to be used in the poultry control of the disease. But if you see these strains, it is slightly, as the ICPI is very high, that is almost 1.3 to 1.4, it started causing, it cannot be used in the early stage, less than 8 weeks or something. It can cause outbreaks in unvaccinated flock if it is this thing. So that is why, because of these reactions, they started working on developing a milder strain. But it had a good protection in the later stage when if it is used in an immune bird with a proper immunity, that can give a better immune response. So when the demand for safer vaccine increased, the lentogenic strains were derived. The first strain which was identified was by Hitchner. It was by accident, I can say. See, when he had some project on Newcastle disease virus identification of as a vaccine strains, he collected some 8 isolates from Budet, who had almost 107 isolates. He gave 8 strains of ND to identify some of the live vaccines, milder strains. So in that strain, he also gave one more isolate of IB as a control group. But by accidentally, the strain which was given as IB for him, when he was using in day-old chicks, it didn't cause any lesions, but it didn't have any HA property, hemagglutination property. But it was found to be a Newcastle disease virus, which even today, even this thing, the old data, if you take, Budet never accepted that this was by, he was blaming Budet, Hitchner, that it was a contamination done in the lab by Hitchner. But there is still that controversy is going on, but that he derived a milder strain which can be used on day old. That is why it is called as B1. B stands for bronchitis virus, actually, which was used as a control in his experiments. So when this virus was derived, it was used on the day old with several flocks, and it had a good immunity during that particular time in protection of this thing. In parallel, when this strain was derived, Budet also studied his whatever the bank he had with the 105 isolates. He screened all those things and derived 3 different strains, which was of milder this thing. From that, the mildest strain was found to be Lasota, which was giving less mortality and less lesions. So that's why he collected that particular strain, Lasota, to be used as a vaccine. So that Lasota was derived from Adam Lasota's farm. That is the name of the farm which this isolate was collected. So that is how the Lasota came into picture. The next is the aspirin strain by England. That was called as the F strain, which was derived. These were the mild strain, but as they derived, still they had some initial pathogenicity in these particular strains which was used in the very initial stage. Then people started working on the cloned vaccines to identify, because these viruses had several quasi-species prevalent in this particular isolates. So they started identifying some of the clones of these viruses and started studying, and they derived some of the strains which has a low vaccinal reactions with a better immunogenicity. That is how some of the strains have been identified, like the clone 30 or the CL79 or the master clone C2. It had its own advantages and disadvantages also in this strain. The master clone is one of the strain from India, which has been identified as the clone vaccine from the Lasota strains. So this is how the things happened, and the next generation vaccines is the recombinant vaccines, which I would like to throw some light on these particular strains, where several of the people are working on this particular strain. They have taken the fusion protein gene from the Lasota, and they have put this particular isolates in the HVT vector, that is the Marek's disease virus genome. They have put this particular gene, the F protein gene, and they are trying to use this as a vaccine and express this protein to be giving a protection against the Newcastle disease. But particular strains or these vector vaccines are more useful, and it has given good results where the virulence of the ND is very less. Like in US, they don't even use Lasota, which causes the outbreaks. And Lasota strains they have the ICPA values as vaccines less than 0.2. So milder strains like B1 or the F strain they use in those countries. Those countries where there is no much big problem with the Newcastle, these strains had a very good impact in control of the Newcastle. That was not an important disease in those countries. But in the Asian countries, when it is very important that we have velogenic Newcastle disease prevalent. That is where this particular strains have some limitations with these things. So if you take some of the points which I would like to share about this, the Newcastle disease has 2 major important proteins in the protection. That is the immunogenic proteins, the fusion and the HN protein 2. We are screening our antibodies and correlating with the protection based on the hemagglutinin neuraminidase proteins, HN gene, HA gene especially, not with the hemagglutinin gene, not with the fusion protein gene. Fusion protein gene is used as a classification of the virulence, and that is also immunogenic. That is also important, but these 2 can give a better protection if it is there. That is what we need to work. And the latency with the Marix virus. If you see, when you use the Marix virus as a vaccine, the Marix virus goes into latency. For continuous stimulation of the antibodies, the virus has to replicate. That question has to be addressed in the forum, that majority of the people has to address this, how to keep the Marix virus replicating and deriving this particular expression of this protein to maintain a good titers for protection of the Newcastle disease. And all the companies with this recombinant vaccines are claiming to have a better protection with the live and killed combinations, which has to be addressed in this particular topic. So overall, it is whether this is of not much use in endemic countries where velogenic Newcastle disease is prevalent for early protection or the long-lived birds. So there we need to address in a proper way. So when it comes to the goals of Newcastle disease vaccines, what should be the goals with the vaccine by vaccinating a flock? What we expect? If you see, if you are immunizing a flock with the Newcastle disease vaccines, whatever, with the live or killed or whatever it is, when there is an infection with the virulent viruses like genotype 13, definitely we expect that to have resistance to infection and reduce the clinical complications like the mortalities or the egg production drop or the growth retardation, ununiformity, all those things. We expect the flock to be immunized in all these ways. Also, we expect all the flocks to have a sterile immunity, that when there is an infection, the virus should not replicate. That is what we expect. But in majority of the time with the velogenic, which is circulating, there will be shedding of the viruses and it will be a continuous challenge, increasing the challenge dose. That is what happens in majority of the time with whatever the vaccines which has been derived. Even if you take any of the pharmacopoeias, they will not address this particular issue with the virus shedding, preventing the virus shedding happening and increasing the challenge viruses dose. None of the vaccines has been. All the vaccines has been tested to protect against the challenge to the clinical signs and the mortality or the production drops. None of the vaccines have been tested or the screen for the shedding of this virus, which has to be addressed. But this there is a lot of data available in the several countries where homologous vaccines, if it is 7i, we have to use 7i. If it is 13, we have to use 13 to have a very good homologous protection to prevent the shedding of the viruses into the environment and leading to more challenges. That is what we need to, that should be the target. Let us try to understand how this immunizations can, the antibody titers to these immunization can protect. Always we correlate that HI titers are very important in the protection of Newcastle disease, even in the influenza viruses also. This is what we derive. We can definitely correlate the influenzas with the Newcastle disease protection based on the antigens used in the test. That's how. If you see the herd immunity, if you take a flock of 10,000, you can't expect all the birds to have a titer of 128 or 256 or 512, which is the optimum titers which can protect against the clinical signs, mortality, and the egg production drops. There is something which is optimum to protect all these things. That is, if it is above 128. So what happens in majority of the flock, you cannot, there will be some birds which is having a low titers, and there will be definitely high titer virus birds also in a flock. If you see the protection and the correlation with the HI titers correlation with the protection, if it is a low GMT, that is less than 1, if there is a challenge, 100% mortality can happen. If the titers are around 8 to 16, the mortality may be around 10 to 15%. If it is more than 5, that is above 32 or the HI titers, definitely that can protect against the clinical signs and mortality. Okay, so that is where I have marked it. If it is above 64, all these birds are protected against the clinical signs and mortality. But these birds, that is above 6.5 to this thing, when there is a challenge, that birds can have production drops around 20%. Those birds which are having a titer of 64 to 128 can have a production drop around 20% in those birds. Okay, so if it is above that, the production drops can be minimized if the titers are high. That is what we expect. So this is what we in a flock we can expect. Most of the titers should fall in the range of 128 to 512. That is what is the optimum to protect against all these clinical parameters. But whether we can derive that is a big challenge in a flock. When there is a challenge to this particular flock, the birds in this range of titers definitely can have clinical signs like the lesions and can have mortality too. But birds above 64, there may not be mortality, but you can expect the production drops to happen. Okay, this is the titers where you can limit the production drop and the viral shedding can be limited by 256. That is what happens. But if you see, when there is a challenge, do you think these birds with 64, they don't die? They show some production drops in the laying flock and some clinical signs in the these things. But these birds recover very fast by developing antibodies with the whatever the plasma cells release more antibodies in the system, and the antibodies increases and it goes up to 4,000 or above to increase the antibodies level in the bird, and those antibody titers can eliminate the virus out, and the birds will come back to the same titer. After once the disease is gone, the titers come back to 64 level. So this is in most of the case in long-lived birds, it has been because of the killed vaccines which happen, the titers are maintained. If you see, this is one of the example in one of the vaccinated flock. It was a 32-week-old flock where the production was 93, commercially flock. Okay, this is what the standard we expect, that is the blue, the bell-shaped curve. So in that particular flock at that particular time, there was high titers of around 2,000 to 4,000. That was the titers. There was an exposure, but the production drop was not present because the titers were optimum, which were able to prevent the production drops. But the titers have gone up because in order to eliminate the virus out from the body, the immunity has responded to increase the antibodies levels to eliminate the virus out. If you check the same flock after 3 weeks, the post-challenge or post-exposure, you can see the antibody titers have come back to the normal range. You can see this is the expected blue one, and this is the normal range which has happened. So this is how the immunity will work in order to eliminate the viruses out. So this is some of the flock where we had monitored the breeder flocks. Okay, so this is the standard what we expect, that is with the blue. Okay, most of the titers we expect to be in the range of 128 to 512. So that is what we expect. If you see, by 21 weeks where there was no pre-lay vaccination done, the titers majority was having 64. Okay, when it is vaccinated at the age of 27, after the immunity has boosted, you can see the titers have shifted to the right with high titers. Most of the titers are falling in the range of 512, but it will not sustain for even more than 15 to 20 weeks with the same titers. We have checked the titers by 39 weeks. You can see there is a shift of this particular titers to the left, where there are titers from 64 to 128 is the maximum. Okay, there are birds with 256 also, but majority of the birds are falling in the range of, this is the time where we need to give a booster dose so that you maintain a good antibody titers. So that is how we have to interpret these particular titers and the protection. And here I also would like to address the issues, the misconceptions which happen with the long-lasting immunity. So if you see, if you take this as a flock, the first vaccination and the booster vaccination prelay, if you check out this, the titers has to go up after the booster inactivated vaccines and has to maintain. If you see the CV is less here, which is a more uniform titers, which goes on increasing once the inactivated vaccine is exhausted. So most of the time, with people are going by boosting with the live vaccines at different interval time. Whether that is going to cause any increase in the immunity is a big question, because those live vaccines doesn't give a long-lasting immunity. It will, if it is immunized already, it will throw the virus out. So it's always better to go for an inactivated vaccine when the titers are coming down below the protective level. So the other thing I would like to address here is about the route of inoculation. The subcutaneous route will have a better holding capacity compared to the intramuscular route. In the intramuscular route, you can get high titers in the very short time, but it cannot sustain for longer periods. But in subcutaneous, you get the immunity at a slower pace, but it sustains for longer period. This is what we need to understand. The next is concepts for earlier protection. We are talking about the high titers above 64 and all. Whether we can achieve in the chicks in the early age less than 6 weeks is a big challenge, especially when there may be maternal antibodies from the parent to the chicks. But when we are placing in a farm, if you see in a farm, there is a brooding shed here, there is a laying shed here. There is a lot of shedding happening with the into the environment. In the early exposure, they can get to the early exposure to the chicks before the immunity is developed. Maternal antibodies can initially protect against the disease to a certain extent, but it cannot give a complete immunity in the early stage, the maternal antibodies. So we have to have a early protection, and this is a big challenge in a multi-age group with less downtime also. So what is the concept we need to understand? There is no correlation with the HI titers and protection in this chicks and the early stage. You cannot expect that. Okay, so definitely live vaccines are very important to develop a mucosal immunity. The same thing happens with the recombinant vaccine also. In the early stage, the immunity will start developing after 30 days in the recombinant vaccines, where there is the local immunity is not there. So we have to achieve a local immunity with the live vaccines in the early stage, in the check stage. So that's why we go for live vaccines in the early stage. But when there is a challenge, when they are not able to, the local immunity cannot prevent, when the viruses enter the system, the antibodies should be ready. But in the chicks, it is more of a maternal antibodies. It can neutralize and prevent to a certain extent, but it cannot have increase the, in the sense, it will not have plasma cells to release more antibodies. The immunity is not well developed in those chicks. So that's why if you have a earlier inactivated vaccines, you can start stimulating the immune response to develop the immunity in the systemic phase. So the live killed combination has been proven to have a better immunity in the early stage protection. Okay, so this is, I will explain, that is how we call as it prevents based on the bottom out effect. How it has been derived, I will try to explain with this particular graph. You see, this is the GMT titers with the high titers of the maternal antibody response in the early stage. Okay, so you can expect high titers even at the 5th day when the yolk is completely absorbed. The titers you can even go high also compared to the day old. So, but it wane offs, it will take around 3 to 4 weeks to completely wane off the maternal antibody response, depending on the immunity. Okay, depending on the antibody titers transferred to the chick. If you are using a live vaccines in the early stage, it will not immediately increase the antibody levels. Even if you are using the inactivated vaccines in the day-old hatchery, you cannot expect the titers to go high. It is not possible. because the immunity takes some time for the maturation and development in the of antibodies. Okay, so definitely the live killed combination, if you take the live killed combination, can give a early immune response compared to only live vaccines, which has been shown in this particular data, which we have a lot of data available in our broiler studies and even the breeder studies also. But definitely in any of these groups, this is a very critical window. When there is a challenge during this particular time, definitely the immunity is not completely matured, or they will not have a high antibody titers in the systemic circulation. You may have local immunity, but systemic circulation may not be there. So this is how the protection may be there with some of the local immunity to prevent the infections. But definitely the challenge is, if the challenge is very high, this is a critical window. So, but when there is a challenge in different age groups, you can see the maternal antibodies can, to a certain extent, can help in these cases. And this here, the active immunity can help. But this is a critical window when the challenge happens. But in this critical window, when you're properly vaccinated also with the live or killed vaccine, when there is immunosuppression, you cannot expect the titers to go high, especially when there is a bursal damage with IBD. When such sort of things happen, some of the even some of the toxicity which happens, which damages the bursa, the B cell immunity will be lost, humoral immunity will be lost. So where you can't expect the titers to go high, the titers remain low, and it will be, the birds will be susceptible to the challenges. So this is what we need to understand in the challenges which is happening. If there is compromise in the feed quality with a lot of toxins happening, definitely the immunosuppressions lead to increase in the challenges. Even if you use live, when there is immunosuppression, if you use live vaccines, it can lead to vaccinal reactions and outbreaks of NDV. Okay, so this is what happens. This is some of the schedule which is followed in India, and it gives a better protection, which according to our data, whatever we have done with our breeders, or this thing which I have been shown. If you see, by the 5th or 6th day, we can go with the live vaccines by intraocular route with the B1 or Lasota, depending on the clinical distinct picture, and with the killed vaccines on the 5th or 6th day itself. The same day you can go for eye drop and go for a killed vaccine injection. So then you can boost it with the live Lasota by intraocular route, it will be the better. But if it is drinking water also, that is fine on the 25th day. And then the 10th week with R2B, then the pre-lay killed vaccines with a 4-way combined, and mid-lay by 39 to 40 weeks, you go for a booster, and every 18 to 20 weeks, you need to repeat if you are keeping for a longer period. This is how the good protection can happen. If there is a high challenge in the flocks, definitely you can include on the 4th week with the ND killed vaccine, and we can repeat before going into lay, that is by the 24th or 25th week in the breeders, you can repeat one more killed vaccine, which can, and no live vaccines during lay. That is what we strictly avoid during this thing. That is how we can get a very good protection in the breeders. When it comes to commercial layers, very similar, but the killed vaccines in the pre-lay goes much early by the 17th week. If it is a high challenge, sometimes you need to repeat the killed vaccines by the 20th week. That is high challenge areas only, otherwise no. And also the one more thing which I would like to address here is the whatever the 10th week which we are giving with the R2B, if there is a lot of immunosuppression cases and a lot of the body weights are not good in the commercial layers, where there is more reactions with the R2B, we can definitely go with the ND killed. Majority of the farms in commercial layers in India go with the ND killed on the 10th week to get a better protection. So that is how we control the Newcastle disease in a better way. Okay, so stop repeating live vaccines during lay. And this is the schedule for the broilers Okay, in a normal, we go for a live vaccines on the 5th day and the 18th or 20th day, with depending on the reactions. If it is a lot of reactions are happening with in a mycoplasma positive flock or some things, we go with the milder strains of ND, that is B1. But if the Lasotas are giving a better protection compared to the B1, so we have to have the Lasota type of strains in this. But that is how the normally we schedule it. But in the high challenge area and in some seasons, like in summer, we have more ND outbreaks where majority of the farms go with the ND killed on day old at hatchery. Day old hatchery, we go with the ND killed, then live vaccines by the 5th day, then booster by the 18th to 20th day, we go with the booster vaccine. So this is what is the schedule which majority of the farmers, majority, I'm not saying 100%, this is how it is the best vaccine which is suitable. That is day old IBH120 with the ND killed. On the 5th day, either with the B1 or the clone vaccines, we can go with the milder strain in the early stage. And then 12th day with the plus vaccine, IBD plus vaccine, and 18th day, go with the ND Lasota, or if IB is a problem, then go with the ND plus IB live. So that is how the schedules depending on the challenges. Wherever there is IBH challenges, we go with the ND plus IBH killed vaccines. That is how the things are happening in our country, which gives a better protection. So definitely the other parameters also are very important, like the strict biosecurity, disposal of the litter material from the earlier infections. All those things matter a lot. Okay, so how much vaccines is important, and how much, whether majority of the time it is the vaccine failure or the vaccination failure which is important. According to me, 90% of the time, it is not the vaccine failure. It is the vaccination failure. The route of vaccination, or the not properly vaccinating by either the eye drop route or the drinking water route, causes majority of the immunity compromise in the broilers or this thing to happen. So let us try to understand what is this vaccinal reactions when majority, according to me, in broilers, ND outbreaks do happen, where we can have high mortalities, like 100% of the flock, where velogenic ND can create 100% of the flocks to mortality can happen. But according to me, 50% to 60% of the cases, it is due to the vaccinal reactions which happen. So the vaccinal reactions means, like the lentogenic viruses which use as the live vaccines, can create more clinical signs like the mild respiratory disease, air sacculitis, production drops in the laying, or the eggshell changes in the laying of these signs, where there is MG or MS positive flocks, that is Mycoplasma gallisepticum or Mycoplasma synoviae. When there is vertical transmission to the broilers, it can create more vaccinal reactions. Whenever you do live vaccines, it can create more problems. And immunocompromised, that is with other diseases like IB or the LP or the influenzas, when there is those diseases are happening, if you give this lentogenic, it can flare up the infections. If there is LP, low pathogenic avian influenza viruses happening in the flock, if you give ND live vaccines like Lasota, the mortalities can shoot up more than 50%. That has happened, and that can happen. So even with the IB cases, infectious bronchitis viruses are causing more respiratory problem, and you give these things can flare up the vaccinal reactions. So we have to be, and even spray vaccinations where uniformity of vaccinations is not there, the cycling of these viruses can cause more problems. And large amount of ammonia or dust can cause more upper respiratory infections. Last but not the least, even the immunosuppression with IBD cases or certain feed cases can cause more vaccinal reactions. Okay, this is one of the example in the laying flock where they have properly immunized with all the live killed combinations in its lifetime. When it check the titers at 28 weeks, the titers were hardly 32 to 64. That is the maximum titers they were getting in those flocks, which when there is a challenge, definitely it can create more mortality and more production drops. But here the production drops was around 30%, less than 30% to the compared to the standard. So when we visited the flock, check the conditions of the management with a lot of complications, or the bacterial infections can lead to more aggravations and the bird can deplete. So even the bad management with poor quality feed can increase the bacterial infections and the fungal infections, and definitely the toxins with the mycotoxins, immunosuppression can lead to suppression of the antibody titers to ND. Any stress conditions can, that for that matter. So this is what happens. And last but not the least, the parasitic infestations. In the cage systems also, I have found several of the roundworm infestation, the Heterachus gallinarum. So you can see, this is one of the video where we took in the cecal worms. It can even enter the oviducts also, these particular worms, and can be found in the eggs also, such sort of parasites and the tapeworms. All those things can compromise your immunity to the ND vaccinations, which we need to take care. So definitely coming to the end of this particular thing, when there is an outbreak, we have to introspect in all these aspects, whether we are in the right way controlling this particular diseases. So when it comes to the host level, the source of bird, age of bird, the metal antibody status, whether it is immunocompromised, all those things, and the vaccinations, vaccination procedures, all those things, vaccination strains, all those things matter. And the agent, how to control the agent by proper cleaning and disinfection. Definitely, according to me, it is more of a 99% cleaning and 1% disinfection, which matters. If you're not cleaning properly, the source of infections you are not removing. Okay. Last but not the least, environment. We talk about biosecurity to in several forums and all, but when it comes to practice, there is a lot of compromise in the dead bird disposals and the litter disposals. This is where I always stress. Majority of the places it has been compromised with the dead bird disposals or the disposals of the mortalities and the fecal disposals, where the viruses can circulate more. So definitely all these things has to be taken care. So this is some of the key messages. Know the right genotypes prevalent in the country and target that things, especially with the genotype 13 presently circulating more. And we need to track if there is any genotype 7i which is circulating in some of our countries. We should be very careful about that, if at all, because most of our neighboring countries like the Indonesia or the Pakistan or the Israel has genotype 7i, which is causing more problems there. And target herd immunity. Definitely we can reduce the shedding into the environment. Early protection to bottom out, to prevent, to have a bottom out effect. Okay. So stop indiscriminate use of live vaccines, which can lead to more vaccinal reactions in immunocompromised things, which can lead to ND lesions, which always will be confused with the velogenic types. Okay. So comprehensive prophylactic vaccinations, proper bird disposal and the litter disposal. Last but not the least, well-executed surveillance programs and the right diagnosis to differentiate between different diseases, which has been presently circulating around the country and our neighbors. So thank you very much. With some food for thought, I would like to give some points. He who does not doubt will never investigate, and he who does not investigate does not perceive the things in a proper way. And he who does not perceive remains in blindness and error. So we should always try to investigate, think in a different way, out of the box, and pursue certain things, which is which in my present practical experience, I have try to do certain things out of the box to identify certain things which was not perceived at all. Okay, so otherwise we will be conducting error if you are compromising with these things. Thank you very much for your